Thank you, Ben, and welcome everyone. So I just want to do official welcome to the second in a series of 2 for the KBase for Educators Workshop. This second one is building off of the first. So if you go into the Q&A document afterwards and you want a refresher on the first workshop, feel free to go to the link for the YouTube there. If you are also unfamiliar with KBase, we have an introduction to KBase on our YouTube page as well. So again, to get it started, my name is Ellen Dow, and I am part of the user engagement team at the U.S. Department of Energy Systems Biology Knowledge Base, or KBase, and I am working out of Berkeley Lab. Joining me today are a few members of our KBase Educators Working Group, including Steve Biller at Wellesley College and Tim Paustian at the University of Wisconsin-Madison. We also have some educators who are helping with the Q&A along with some of the KBase team, so please drop your questions in the question and answer document, and we will be answering them as this webinar proceeds. And we will have some time at the end to go through those questions as well. So in the second workshop in the series, we're going to introduce some more resources and how we are providing support on adapting them to your courses. And so again, this workshop series is a little less formal than our typical webinars. So please ask your questions in the Q&A doc, and we will have time to answer those at the end. So while I go through our agenda, feel free to log into the narrative. If you would like to follow along, we'll be sharing some of our narratives, which are linked in that Q&A doc as well. So this the second workshop builds off the first, so I will go over the concept workflows and give an overview of the different concepts that we have that are built out. And then Steve will go through the genome analysis workflow, and Tim will go through the metabolic modeling workflow, which is new. We just finished that at the beginning of the summer, so we're really excited to share those. And with that, I'm going to launch into the more formal version of, or into the showing everybody what resources we have. So remember, if you want to follow along, go into the narratives. So just log into KBase and go to our orgs. If you have not yet joined our org, click on the orgs. You can search for KBase Educators. I belong to this group, so it shows up for me, and make sure that you have all orgs selected in case you do not belong. And then once you click on the org itself, you will be able to request to join. And we have Zach who's standing by, or I can also have some other members let you in, or we'll let you in after, we will accept your request after this webinar. To get started, I wanted to go through the concept workflows themselves and the different topics we have built out. So these are all built out from our educator working groups. So these are different groups of our educators that started last summer, and we've actually continued to work together to build out these different concepts. And these include genomics, metagenomics, phylogenetics, pangenomes, as well as metabolic modeling, which is the first different topic. And so each of these workflows are broken down into modules that adapts the narrative. So the narrative interface itself, we've used each narrative to translate as a single module. And each of these modules addresses a different topic within the concept workflow. And it also embeds a few different pieces for different, it embeds some information about background, as well as instructions, including data, different apps, as well as embedded questions. And each of our educators today will walk through how those have been adapted. And so within this concept workflow, which acts like a landing page for these different topics, You can actually click on the different links here for each of the different concept workflows themselves, and they will take you to those individual pages. Within the concept overview, there is also different resources for a few different narratives, including the module skeleton, as well as supporting documentation. So if you remember, if you were here the first time or listened to the first workshop, this is where we have best practices as well as a documentation site. There is a link as well to the KBase YouTube channel, which includes some great resources for how to with different workflows, as well as an introduction to KBase itself. And so I wanted to walk through how each of these workflows have been built out in general. So following the KBase module skeleton, how these different workflows have been broken down are through including information on scientific background. And then we go through sample processing, especially for the first workflow, if we introduce new data, introduces the data inputs. So there's a lot of helpful information in this module skeleton on, different how to add data to a narrative. And then it goes through the different acts of using an app. So going through and being able to use these apps for data exploration and analysis. And then within the results, when students have results or prompting them, are embedded questions to help prompt students on and going through the apps themselves with the outputs, as well as the different parameters. And we've included some different comprehensive questions within these modules and provided some structure for reflection and setting up for next potential steps in the pipeline. So within each of these narratives, we actually have them linked. So when you scroll down to the bottom of the first narrative or the second, it prompts you to go to the following narrative, which is linked within there. And so with that, we are right on time. I will stop my share and let Steve take it away.
All right, thanks a lot, Ellen. So I'm Steve Biller. I teach in the Department of Biological Sciences at Wellesley. And so I've been using KBase for the past year and a half in my classes. For context, both in a 60 student introductory genetics class or 200 level genetics class, as well as in an advanced undergraduate seminar in bioinformatics. And so this genome exploration module is one that I started using last year in genetics when we were teaching fully remotely due to COVID. In sort of a 2.5 hour exercise. So had students working together in either pairs or groups of 3 in Zoom breakout rooms to kind of go through this material. But we're planning on doing the same module again, sort of now that we're back for in-person labs as well. So this genome exploration module sort of comes after a section of the lecture portion of the class where I sort of introduce the concept of how we sequence genomes, how we interpret genomes, and give sort of very high level overview about how that works. That's more in the context of kind of more human genomes. But then we also talk about, the idea of horizontal gene transfer in microbes. And so this module is sort of designed to kind of combine a lot of those concepts and put it in a format that's kind of tractable, where the students can actually see how all of this works. Again, this genome exploration module is sort of broken up into different sections, with kind of going at different parts of the process of assembling and interpreting a genome. And you can customize it however you want based on what's relevant for your class. So for my advanced course, I use a version of this where the students are doing more things on their own, running more of the analyses on their own, and kind of give them a little more kind of freedom to explore and analyze their own data after they've gone through this type of a narrative example. In the 200 level genetics class, we only had 2.5 hours. So we really focused more on, interpreting the data rather than kind of running the analyses. So I left more of these, this module with kind of pre-filled results that had already been computed so that they weren't kind of, using the limited time we have to get it to run. Some things like the actual assembly can take some amount of time. But the focus was really on sort of clicking around and exploring with sort of guided questions, what's going on? How does all this kind of relate to what we talked about in the class? This first part of this genome exploration module is, again, sort of following this structure that Ellen gave you an overview of. And the idea of this is it really goes through an example of looking at a methicillin-resistant Staph aureus genome. Antibiotic resistance is something that certainly grabs the attention of our undergraduates. It's a great example for thinking about these concepts of horizontal gene transfer, and it gives them something sort of tractable that they can go in and kind of search for and explore these genomes. So the overall kind of goal of this is to assemble this genome, annotate it, and then figure out what you can learn about what some of the genetic underpinnings of this antibiotic resistance phenotype might be. Where can we go with just bioinformatics? And then bringing that back out to, okay, now what else would we need to do experimentally to sort of verify that? So we start with, there are some very basic overviews of the concept of shotgun sequencing and how we assemble a large genome from short individual pieces, what it means to be a forward or reverse read from an Illumina technology. Again, all these data sets I had left sort of already loaded in, but depending on what works best for your context, you could let your students do all of this on their own and actually kind of go through all of these processes from the beginning. So first they import all of the raw data from the NCBI sequence read archive. And then I have them look at assessing the read quality using FastQC, where they're looking at these plots visually about what a base score means, and it's sort of an opportunity. I would sort of have them go through a few steps, and then we'd all there are places where I'd say, okay, get to this step, think about these questions, and then we're going to come back as a group and talk about what this actually means, and make sure as we're kind of working our way through this whole workflow, that everyone is sort of on board and at the sort of the same place in this narrative. So we do some data quality control, have them go back to think about, in class questions like this about, think, okay, well, now after we've trimmed low quality regions, how many reads are left? So this is both to kind of get students to just talk to each other and try and figure these things out, and really just encourage them to click around and explore and see, what they can find in all of this. So have them go through this trimming. We have them reanalyze, what the quality of the data looks like after that and how many reads are left. We talk about why, some data is good and some sequence data might not be so good, and why we would want to remove that from an assembly before we do that. Next, we go into the actual genome assembly step. So talk at a very, very basic level about the idea of a De Bruijn graph or Kamer-based assembler. Think about how these individual reads can be broken apart and stitched together to make a final read, and sort of what that means for assembling the whole genome. And then I have them compare two different assemblers. So if you take the same data set, into IDBAUD versus into SPADES, what is the difference? Is it the same? Sort of the idea for some students that you can take the same data set, give it to two different algorithms and to get two different answers kind of, is shocking and surprising to a lot of them. And I think an important lesson that the methods matter, and that how you go about answering your question has to be appropriate to what you're actually doing, what kind of data you're dealing with. So we have them compare, the IDBA UD assembly versus the Spades assembly. Think about, okay, what is a contig? What does this sort of vocabulary mean? What does it mean to have, a largest contig and total length? Is one of these genomes right or wrong? What does it mean to be a draft genome versus a complete genome? These are all sort of questions I have them answer in class, and then we have a small group discussion about that. There are also comprehension questions at the end of each of these modules, which are questions that I had students basically go through and turn in as homework after the lab. So slightly, high order, more, kind of questions to really kind of check and make sure that they actually understand what all of this means and how to sort of apply what we've been talking about. So I just have them, submit a couple sentence responses to each of these. So after we've assembled the genome, now we move on to the next part, which is annotating it. So this is an opportunity where, I talk about, what we had discussed in class about, what makes a gene, what are sort of some of these basic properties of a gene, a start codon, what's a stop codon, what does it mean to have a ribosome binding site, what are promoters, all of that. So sort of use this diagram as a place to begin discussing that. Think about the basics of, how do we go about annotating it? How do we turn this, just long stretch of A's, T's, G's, and C's that we got from that part one and actually make it something that it is kind of biologically interpretable to kind of mere mortals like us. So I have them annotate the assemblies. Again, this is something that I had pre-filled for my introductory classes. For my more advanced classes, I kind of let them run with this on their own. We have them go through and, go through the features and look for, okay, what is it in there? What can we kind of see? What have we learned about this Staph aureus strain just from this annotation? How many protein coding genes are there? Where would you find DNA A? So I have them just search through there just to kind of, find it and kind of see, okay, well, what does this number mean? How do I actually interpret that? And then similarly, a comprehensive question at the end. So at this point, as you saw in the overview diagram, we sort of have them branch a little bit to sort of, thinking about, well, how do we actually go, coming back to our original question, which is what is the potential genetic basis for antibiotic resistance in this organism? We think about, well, what can we learn from just thinking about this genome on its own? And then in part 4, sort of take more of a comparative genomics approach and say, well, okay, well, what can we learn sort of from this type of it, more of a pangenome-based analysis versus a single genome analysis. So if I jump to the single genome analysis module, so here, we've now assembled and annotated this genome. We do a genome visualization, kind of one of these classic circle plots, which, for the question of figuring out antibiotic resistance is not necessarily all that helpful directly, but anything with sort of, the ability to sort of have these visuals and these graphics is really helpful to the students to think about, okay, well, here's all these dots. Now, what does this actually mean? Okay, what is a CDS? What is a tRNA? What is an rRNA? So it's both a time to go over kind of those concepts as well as the terminology again. Why do we care about GC content and GC skew in terms of, things like looking for deviations related to potentially to horizontal gene transfer events and things like that. The ability of KBase to sort of have these more interactive visualizations found very, very helpful. So I have them actually search through the features of the genome to look for things related to antibiotic resistance. I don't necessarily directly tell them right off the bat how to do that and let them try and figure this out. But there are certainly some genes that are annotated as antibiotic resistance proteins. Most students sort of figure that out after a few minutes. They go about sort of different ways to think about, okay, well, now how am I going to go about doing this? How am I going to figure out what this actually is? We also, For some classes, I have them actually look at sort of metabolic reconstructions. Tim's going to talk to you about a much more detailed and involved way of sort of going about doing this than what we were doing with just kind of recreating it based on the MRSA genome. But this is also an opportunity for me to kind of have them go explore what does it mean to have a pathway. What is this pathway? We have a number of students in the biochemistry program taking our genetics class, so this is sort of a way to start relating some of those concepts as well. And again, any of these things that aren't relevant to your class, you can also just remove. The last part here is to, rather than looking at annotations, and I talk about sort of the limitations of annotations, but I also have them, blast some different kind of known beta-lactam resistance genes against their genome. So I give them the FASTA sequence of a few different examples, and I ask each group just to pick one randomly, and then do a protein blast against the feature set for this genome, and they will find homologs in the genome, which they get very excited about, and then then we sort of go, okay, now what does that mean? What does that do? Where is it? And that sort of thing. So they get to see sort of a couple of different approaches for how we would go about trying to do this. And the last part I'll just quickly go through is really thinking about more of a comparative approach. So kind of the idea of, well, we can learn some things from one genome, but there's certainly a limitation to what we can do with annotation. But what if we sort of have two pretty closely related genomes and we compare them, and we can actually look for, what's the same and what's different between them, and take more of that approach? Maybe that would be a different way to think about what some candidate genes might be conferring antibiotic resistance. Just because we found those homologs by blast doesn't guarantee that those are, the actual, functional genes necessarily. So I first have them, compute in a comparison of the genomes based on an average nucleotide identity metric, and sort of explain what that is. Have them compare the proteomes with these sort of kind of classic dot plot kind of visualizations, where you can sort of see, some regions of local synteny of genes, but also gives me a chance to discuss, sort of recombination and variation in genomes between these two Staph aureus strain, one that's methicillin resistant and one that's methicillin sensitive. And then kind of at the end, have them think about this idea of a core versus pan genome in microbial genomes. So which genes are sort of common and shared with both this resistant and this sensitive strain, and which ones are sort of exclusive to one genome or the other, and thinking about how you use that as a tool for looking for kind of the genetic basis for this phenotype of antibiotic resistance that we're interested in. So it takes them through kind of these steps. Again, most of this I had pre-computed for them. And then we go through and kind of talk about how to go through the data that they're getting back to analyze, well, if it's in two genomes, okay, does that mean it's in the core versus the pan genome, and kind of reinforce all of these ideas. And then some wrap-up comprehension questions. So again, it worked pretty well, in our first trial as a kind of 2.5 hour-ish interactive exercise. Tried to, because we were in kind of remote world, I did a lot of having students come in and out of their breakout rooms, just to make sure we were all talking about it, make sure that nobody was just kind of just copying and pasting things, but we had a chance to really discuss these concepts. But it the feedback from this was really strong. A lot of students really enjoyed kind of the visual aspects of KBase, being able to actually kind of work with it from a instructor perspective. And I think Tim will probably echo this. It was nice to be able to do this for a large number of students without setting up a computational cluster or having to do any command line work, especially with beginning undergraduates, many of whom had basically no computational experience before this, and they got to really dive into analyzing whole genomes. That was a lot of them really enjoyed that lab. So we're going to keep going with it in the class, and keep kind of adapting it, and hopefully we'll develop some additional modules or other examples to use. But it's been a great teaching tool for us so far. I'll stop there.
Okay, I think it's time for me to take over, and I will try and do that. All right, so first of all, before I say anything, I just want to say that KBase has been just a wonderful tool to use in our program. We originally were using things like Mother, great software, it's in the community, but we found the same things that Steve did, is that the students just, the command line, getting the commands right, typing things in, variations in computers, et cetera, just made this cognitive overload that they completely missed the overarching concepts we were trying to teach. When I found out about KBase, which someone just offhandedly heard about it and said, "Hey, maybe you should try this," and they hadn't even looked at it, and I found this thing, it was like finding a gold mine. Just because this puts it all in one place and it's really easy to use. So in our case, we had students in 2019, the first time I used KBase, isolating organisms from a deep sea ocean vent. Professor Karthik Anand Theraman does research on deep sea ocean vents, and he was willing to go down and collect samples for us. Then we used some novel isolation methods to see if we could start pulling into culture what Professor Anand Theraman had predicted, and using different media. And so we did that, and then we sequenced, we did preliminary sequences, identified unusual species, and came up with 11 candidates that we sequenced their entire genomes. And that's what we're using now. They had gotten to the end where they sequenced the genomes, they had done some comparing of the genomes, et cetera, in there. But I came into the class last spring, and it's like, okay, we're face to face, but how can we do this safely? What can we do? And I came up with a project of really characterizing these organisms. And I realized that KBase could do all sorts of metabolic modeling. So what we did is we took the sequences first and we assembled them. And this was done outside of KBase. And we go through, say what the goals are, et cetera. And then there's instructor notes. We used an assembler called Canoe. This is Oxford nanopore sequences. And we use that assembler, and it's quite nice. You can use it outside on your laptop, and it will assemble. It'll take about 8 hours if you've got enough memory, but you can do it. And so this explains how to use Canoe, goes through the prompts, et cetera. And so we had them assemble the genomes and talked about what assemblers do, et cetera, et cetera. So then once they got the assemblies, I actually did this for them because, again, we had 11 genomes. Each one took 8 hours. And so therefore I did the assemblies and then gave them the data, but we definitely had them practice with other genomes. And we got that information. And these were 10 isolates from the deep sea. So the goal was to characterize these guys metabolically and then test out those predictions that KBase made in the laboratory. So KBase has all sorts of tools to do this. We went through, got the genome assembly, had them answer some questions about it, and then we moved on to the next thing, which is genome annotation and drafting a metabolic model. All right, so that's going to have to come up. All right, so now we've got the assembled sequences. We brought them in, and this goes through an overview of genome annotation, what it does, how it works, how the algorithms work, and just explaining it in really broad strokes, and then what we're going to use. We also talked about how to import data into KBase. Since these were our own sequences, they're not available to the public, but we're sharing them now with this, through these modules. So these are available to anyone. You can import data in, you can take any FAFSA files that you have or a number of different other types of files and import them into KBase and it will then use them. We then had each group, I had about 50 students. So each group of 4 got their very own microorganism, and that was their organism to work with. And they copied my narrative and then went through and had to do these same things. We used the annotate microbial assembly with RAS-TK. I got output from that. And then, as Steve talked about, we had them go through and answer questions about how many protein coding regions are there, what can it do, taught them how to read through the data. And again, if you go through this, it has just a wonderful interface for finding out what's in there, right? You can go through and search for different things. It'll give you information about it. So then the next step was, once we've got the sequence, we've annotated all the genes, we then want to go through and build a metabolic model. You run that. And these programs actually don't take that long to run. So in a laboratory setting, you can run them and have the students run them. I think we were, if I look at the job, let's see, how long did it take? Ran for 2 minutes and 10 seconds. So it's very quick to run these kind of build metabolic models. And then again, you get these results in this interface. It gives you a report. It will also often give you output. You can go in and look at pathways and start asking questions. Like one of the questions we ask them is, does this organism have a TCA cycle? So it's right here. They can click on it. If it's not there and you say, does it have a histidine metabolism? You can just put in histidine, have the students click on it, and teach them that, yes, you can go through here and look at this and see, does it have all the genes necessary to synthesize histidine, and just by the blue color, et cetera, et cetera. So we have them examine the results, make some predictions about, what it will be able to do, et cetera. And then we say, look, this is really tedious, right? You don't want to go through thousands and thousands of genes and pathways trying to figure out what it can do. So there are computer programs to do that. And one of the programs they have is run model characterization, which it will go through and it will make predictions about what kind of auxotrophies you have, what kind of, what abilities your organism has. And if you run that run model characterization and you look at the output from it, here, it will give you what, oh, I'm sorry, yeah, that's not, yeah, okay, yeah. It will give you predictions. No, okay, I'm sorry. So that's, you get these pathways, but if you go on and use things like predict genome auxotrophies, what's the new one called?
Check model mass balance.
Yeah, and what is it? Check
model mass balance.
No, I don't think that's the one I'm thinking of. It's the one that will predict like amino acid pathways, etc. Anyway, you done. So if you go through and run model characterization, you'll get those answers for you. And then if you, so then they can get. Oh, okay. I'm sorry. Here it is. I'm sorry. I missed this. So if you go here, it'll give you auxotrophy statistics, and will tell you what it's auxotrophic for. So the organism can make predictions about, what things it cannot do. And so if you go through and look at those, you'll see that, for in this instance, it says it's not an auxotrophic for anything, because it seems to have all the biosynthesis pathways in its genome. At least the program can find them. So then what the students did in the classroom is then they took the strains, they brought them into, brought them in culture, and then tested these, created media to test. Okay, can it really grow on mineral media? And in the case of the one that we're running here, which is Marinobacter algicola, right? It turned out that yes, indeed, this was not auxotrophic for anything. It can grow in mineral media. So I had the students do that with all their different narratives. And we actually accumulated these results, and were able to actually determine how accurate KBase was in predicting the capabilities of these organisms. It turned out it's over 90% accurate in its predictions, which is really interesting data for them. There's a whole bunch of other things that you can do, and I'll touch on those a little bit. Okay, so no, I was in the right place. Okay, as if you go on from here and you get to the end, you can then go to gap filling and flux balance analysis. And in this, it will go through and look at, okay, what are there, like just a couple gaps in some of the metabolic pathways? Does that work? How can we get that to do, making educated predictions about exactly what we're going to get, what pathways might be there and what pathways might not. And then in the flux balance analysis, you can simulate the metabolism of the organism and look at the fluxes and see what it would do. And this is all preparatory material for maybe making a prediction about what kind of media can be used, etc. I'm probably, I still got a little bit more time here. So what we found out in our research, in our work, is that, they could do that, but then the students started exploring KBase and they started finding really interesting things. Like there's phylogenetic analysis that they can do where they could take their whole genome, compare it to related genomes and build trees and make predictions about which ones are unique and which ones are not. And we actually found one organism that was unique enough that it was worth getting we had nanopore sequence data, we got the Illumina sequence data, and we're able to assemble a professional quality map just from that data. And so the students were pretty excited about that, and that they had found a novel organism from deep sea ocean vents. So we went from there. As you go through this, you can then talk about, experiments with different media for students and impact the growth of the metabolic model, where you give it different media. It goes from there, talks about the environment of the deep sea ocean vent. This is a very cool crab. And you can go through there, and we actually put in the JW1 media and then what it had, and then they could, make manipulations and see what happens to the flux balance analysis. So this is all explained in here, and you can go into great detail with all this. And then you can then go on to module 4, where if you want, you can compare the different media and see what works, and also compare metabolic models between closely related organisms and learn more about them. Okay, that's basically all I had for this. But again, definitely KBase is something to go through and really play around with and understand, to use. I found when my students were done with it, they had a much better understanding of what we were doing in class, the bioinformatics of the class, and what the tools were actually doing for them. And when we had them give poster presentations at the end of the semester, and the data that they had from those poster presentations and their understanding was worlds better because of KBase and this platform.
Thank you so much, Tim. So before we go to any Q&A and answering any of the questions that we don't have answered yet within the document, I just want to remind you that if you would like to join the KBase Educators community, please send us an email to engage@kbase.us. Sign up for a KBase account or sign into your KBase account. Remember to search all the orgs for the KBase Educators Org and request to join. Send us an email to engage@kbase.us to let us know that you would like to join the org. Please share your username, as well as when and what you're teaching, and if you would like to connect with other educators. We are happy to connect you. And with that, I want to see if we have any questions still to answer. we just got a new one. So Tim, for your class, was this a bioinformatics class or a graduate level genetics class?
Okay, so the class that I taught, this is actually it's our CURE, our classroom undergraduate research experience. It's the capstone class for the undergraduate major. And every year we come up with different projects for them to do. And so I thought this is a really good one, given this, COVID and the circumstances we were in. We were able to do this in the laboratory with the students. And yeah, so it was not just a bioinformatics class. They ended up using a lot of bioinformatics tools to answer their question. And that's by design, because this is one of the places they can get that information in the major. But the real focus was on the research and using these tools to actually do some novel research.
Yeah, this is a really neat example of integrating both wet lab with the bioinformatics analysis itself in KBase. And then I have a question for any of our educators who are on the call, Steve, Tim, Carlos, and Aaron as well. My question is, so it's not always clear what parameters to use for students. So how do you educate your students on how to enter what parameters to use for each different app analysis?
I'll take a stab at it. Basically, KBase has wonderful documentation. Go through and read through the documentation for each model that you're going to run or app that you're going to run, and it will tell you the parameters, what they are, what they're used for. A lot of the time I found you can leave them at the defaults and it works just fine. There's only a few required parameters in a lot of the things, and then it's a matter of figuring out what's actually needed to run your analysis. So a fun, interesting example, I'm actually doing for another class this fall. We're going to be using the modules for transcriptomics and looking at different things there. We're doing this in Rotobacter spheroides, which is a very high GC organism. So when you run quality control on it, you get all sorts of red errors, and it's because it expects the GC balance to be around 50%, and it's not. It's like 67, 68%. Then that's a great teaching moment for the students, in that you got to be aware of what the results and the parameters are. But no, yeah, most times the defaults are really good, and the documentation is fantastic in KBase.
Yeah, I'll just follow up I didn't really worry very much about parameters for my 200 level genetics class. I just basically told everyone we just basically took the defaults. But it is something that I spent a lot more time talking with students about in my 300 level bioinformatics specific course. So kind of similar to what Tim was saying, I had groups of students actually go into the documentation and basically give a quick in-class presentation on some of the different key algorithms and what they did and what some of these parameters were. So they would kind of take some time, figure it out on their own, talk to me about it, and then sort of present it to the class. And I did have some exercises where I would, take certain like genome assemblers and have them actually test, okay, now what if you change, your K-mer length defaults in some of these assemblers? What did that do to your assembly and things like that? And so they could explore a little bit of the parameter space, again, focusing mostly on, the key ones that were most relevant to sort of understanding how the algorithm worked. There's too much of parameter space to optimize all at once in these contexts, but it can absolutely be done.
Thank you. And so I think I have one other question that I really want to ask, and that is from your any of our educators who are here, from your experience of going through these different modules, what is the best way, or how would you now introduce your students to using KBase after going through this process? And more of the basics behind using KBase itself.
I can try and jump in and answer that question. So I used KBase in an undergraduate genomics and proteomics course. And we started it last year when the university was totally remote and everything was online. And I think KBase was really a great tool. It was amazing for that. But the students, the sort of learning curve of understanding what to click and what parameters they needed to just leave as defaults and what could be changed. So I think in the future, if I had to introduce it, I would love to be able to have a small class period where we kind of work on it together, because if I'm looking over their shoulder and just saying, click this, do this, I think with spending maybe a half hour with the students working through some early steps, then because the platform is, once you understand the basics of the apps and how they work, then they can scale up, I think, very, very quickly. And doing that remotely, where I was, okay, share your screen with me now. Now let's look at this. Now click this, do this. That was a little more tedious. I think if we could do it all together, if I had to do it again remotely, I think I would create like a little video where I step through and click through and show them, and that wasn't something that I did the first time. I tried to just address things on a case-by-case basis. But if I could have a little in-person time, I think I would use that to have them go through. And once they're familiar with the apps, then I think they can run with it from there.
Thank you, Aaron.
Yeah, I would also agree with everything Aaron said. It's much easier to do it in person. When you're there, you can, we actually did a little bit of an introductory thing of, okay, let's go through, this is how KBase works. Number one, you guys have great docs for getting started. And then I pointed them to that, but then also in class, we kind of walk through that stuff. But you're absolutely right, Aaron. Once they understand, how the system works, they start exploring, and they actually came up with some pretty innovative ways of doing things.
I love your class, Timothy. That's really cool, and Aaron's emphasis on really getting them up and running, and the documentation is really great. The quick start is really great, but this year that we're doing it again this fall, I think I'm going to spend more time with an intro narrative so that they see just the structure of the narrative. And I really want to promote the help-seeking environment where, okay, our task is to do this, assemble or create a metabolic model. Now that you got me all interested in metabolic models, let's look at the tools that we're using. So look at the info tab and now try to find the paper and why is this assembler different from the other assembler. And we started doing that last time we did this class, but I did not emphasize that enough. And I want to really try to have students discuss both in person this semester, hopefully, as well as on the discussion forums. Why did you pick these default parameters, or why is this tool different from the other? And using the resources that KBase has, as well as linking out to the original papers if possible.
That's a great point, Carlos. And one of the pieces I actually worked on in response to that was I did create a student intro to KBase narrative, which is available within the org. This was also in part creating some activities and putting in some links, such as that quick start guide and some different aspects of documentation from KBase. So we have the YouTube page with the main page linked to the webinar and tutorials, as well as the quick start guide and troubleshooting if there are any challenges that are really difficult to overcome. And I use this activity a bit with some high school students this summer. So I had them go through and add some data to a narrative, run an app, and as well as use Markdown. So we have these text cells within the narrative that you add different questions or answers to if you're a student. And so I had some students play around and work with this, and they seem to be quite adept at learning how to do it with this. But I think it would be really great to actually have a little bit more of a hands-on period with that. And then I also included instructions on actually sharing the narrative so that it provides a bit more background for students to share their narrative themselves with their instructor. And so that is one of the other supporting documentation and narratives that we have. And so with that, I don't think, so yeah, I will add this this narrative to the Google Doc and make sure it is in our Q&A doc before closing out today. I don't think we have any more questions. So I just wanted to thank everybody for coming today. I'm going to share the feedback form, so please let us know how we did. As well, follow us on Twitter at DOE KBase. That's where we share a lot of these different offerings for webinars and such. Also follow our YouTube channel. This is where we post our webinars and different workshops like this one, and different resources for all of our users. And with that, thank you so much, Steve, Tim, Aaron, and Carlos for coming on. I really appreciate your guys' work, especially as educators within our working group and part of our community. So thank you, everyone, and I hope you have a great day.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.